So if you're someone who keeps up with health news, you know that the headlines about Alzheimer's can feel like, well, a total flood. It's honestly overwhelming, and it's tough to know what's just a small step forward versus what might be a real, genuine leap.
I mean, one week you'll see a study talking about a link between, of all things, full-fat cheese and dementia.
And then the next, bam, there's a headline that some hidden compound in aloe vera is the next big thing.
Then you'll get news that a few weeks of brain training could somehow protect you for, like, 20 years.
And of course, there are always those studies telling us that something as simple as going for a walk might just be our best defense. It's a lot to sort through.
So with all this constant noise, how in the world do you spot what a real breakthrough actually looks like? Well, pretty recently, one story in particular managed to cut right through all that static. It's all about something that researchers have started calling a hidden death switch. And yeah, that name is intense for a reason. This wasn't just another headline, it really signaled a major shift in how we might understand the actual mechanics of this disease.
And the announcement itself was just so powerful. A team from Heidelberg University basically said, and I'm quoting here, "Scientists have uncovered a hidden death switch in the brain that may be driving Alzheimer's disease." I mean, a sentence like that, it just grabs everyone's attention, from the top scientists in the field to families who are living with this every single day.
So what is this thing? What is this ticking bomb inside our brain cells? Well, to really get what this death switch is all about, we have to look at one of the key players in our brain's massive communication network. Let's talk about NMDA receptors. These are basically proteins that hang out on the surface of our nerve cells, and they are absolutely crucial for us. You can think of them like docking stations that catch signals flying around. They would allow us to learn things and make new memories. So normally, these guys are definitely our friend. But, and this is a huge but, it turns out their location is everything. See, the helpful pro-survival receptors are located right at the synapse. That's the little connection point where brain cells talk to each other. But there are other NMDA receptors hanging out outside the synapse. And when those get activated, they do the exact opposite. They trigger all sorts of toxic stuff. It's kind of like a security system, right? The sensors on your doors and windows? Those are good. But a random rogue set of sensors somewhere else could trigger a full-on self-destruct sequence.
And that rogue sensor, it turns out, has an accomplice. Researchers found out that this other protein, called TRPM4, actually links up with those bad outside the synapse receptors. And when they join forces, they create what the scientists are calling a toxic death complex. And that complex, that right there, is the death switch. It's the mechanism that literally tells a healthy brain cell to self-destruct.
Okay, so finding the problem is a huge step. But how do you actually fix it? Well, what's so cool is that the very same team that found this death switch also came up with a potential way to disarm it.
They created this custom-designed molecule called FP802. Now, the technical name is a twin F interface inhibitor, which is a mouthful. But basically, it's a molecule that's specifically designed to get right in between those two toxic proteins and just break them up. It's like having the perfect key to unlock and dismantle that deadly partnership.
So here's how it works step by step. First, you've got the harmful receptor and that TRPM4 protein pairing up, forming the death complex. Then, the FP802 molecule is introduced. It zeroes in on the exact spot where those two are connected, their interface. It binds right to that connection point, and by doing so, it physically pries them apart, breaking the bond. And just like that, the death complex is dissolved, and that signal telling the cell to die is stopped in its tracks.
So, the big question, did it actually work? Well, in mouse models of Alzheimer's, the results were pretty stunning. The disease's progression slowed way down. The loss of those vital connections between brain cells was reduced, and here's the most important part. The mice's ability to learn and remember things stayed largely preserved. It even reduced the buildup of those amyloid plaques we always hear about.
I mean, all of this sounds incredibly promising, right? But it brings us to that big elephant in the room question. How do we get from these amazing results in mice to a potential cure for people?
And that question matters so much. Alzheimer's is devastating. As someone with a family history of the disease put it, it's just heartbreaking. You're taking care of somebody who doesn't even remember who you are. The human cost is just immense. And that's why every single one of these potential breakthroughs gets, and should get, so much scrutiny.
Which brings us to the really important caveat. We have to be clear, these were preclinical studies.
in mice. A treatment that you or I could actually use is still a long, long way off. It's going to take years of development, safety testing, and massive clinical trials. A lot of drugs that look great in mice, well, they never make it to the pharmacy. So this is an absolutely critical first step, but it is just the beginning of a very long journey.
But here's why this is still so exciting and why it really is a totally new strategy. For decades, a lot of the research has been focused on cleaning up the mess, clearing out those amyloid plaques after they've already built up. This approach is different. It's proactive. It's not about just mopping up the damage. It's about preventing the damage from happening in the first place by flipping off the switch that kills the nerve cells. It's a huge shift from treating a symptom to actually targeting a root cause.
And you know, it's a profound change in thinking that leaves you with this one kind of wild question. If we can really learn how to turn off a cellular death switch, what other commands could we one day learn to give our own brains to repair them, protect them, and maybe even enhance them?